So cytosine, guanine, adenine, uracil. Define. Deoxyribonucleic acid, chromosome, mitochondrial DNA, viral DNA. So what do they define? Obviously, deoxyribonucleic acid, no, because in DNA there is no uracil. Ribonucleic acid, yes, because in RNA there is the uracil. And they define the chromosome, no. You might be tricked and put yes, but you should focus more on reading the question correctly. Saying define, this means do nothing with the chromosome. Mitochondrial DNA, false. Also, DNA does not contain uracil. Viral DNA, also false, as long as it's DNA, as long as wrong. 2. Okay, RNAs are different from DNA because they contain uracil and ribose, they contain uracil and thymine, they contain uracil and deoxyribose, they contain ribose and thymidine, they contain ribose and deoxyribose. RNAs are different from DNA because we should focus on the question. So RNAs are different. Now we just want to show the difference between RNA and DNA. So they contain uracil and ribose. Yes, exactly. Because DNA contains deoxyribose, which is also different from ribose. DNA contains thymine, however, RNA contains uracil. So here, that's correct. Okay. Contain uracil and deoxyribose? No. RNA does not contain deoxyribose. This false. Oh, we didn't do first one. Contain uracil and thymine? This is also false, because RNA does not contain thymine. It's either uracil or thymine, one of them. Because these two nitrogenous bases, they replace each other's. They contain ribose and thymidine. This is wrong, obviously. There's nothing thymidine in DNA or RNA. They contain ribose and deoxyribose. This is also wrong, because this nucleic acid must be containing either ribose or deoxyribose, not both of them. Perfect. Number 3. Circular DNA is found in mitochondria, in nucleosomes, in bacteria, and nucleolus in the Golgi. So it's obvious that it exists in the mitochondria. Mitochondria has circular DNA because according to the endosymbiotic theory, mitochondria used to be a bacteria, a prokaryote, and then it developed to adapt with other organelles inside the cell. So that's why it's like any bacteria, it used to have circular DNA and it still has its own circular DNA. The circular DNA exists in the nucleosomes? No. Bacteria, obviously. Nucleolus? No. As long as we have a nucleus or a nucleolus, this means we're speaking about a eukaryotic cell. As long as we're speaking about the nucleoid, means we're speaking about a prokaryotic cell. Okay? So nucleosome and the nucleolus, this at all wrong, because both of them, nucleosomes and nucleolus, exist only in the eukaryotic cell. However, circular DNA exists only in the prokaryotic cell. Does circular DNA exist in the Golgi? Obviously no. Okay. DNA in cells is found in the nucleus, in microtubular organizing centers, in mitochondria, in polyribosomes, in the cytoplasm. So DNA cells is found in the nucleus, obviously. Microtubular organizing centers, wrong. Mitochondria, obviously, because mitochondria has cell DNA. It's also double helix, but it's circular, it's not linear. In polyribosomes, this is so wrong. And because usually ribosomes, the subunits of the ribosomes hold RNA, does not hold DNA. And in the cytoplasm, this is also wrong. Okay, DNA does not exist in the cytoplasm. Okay, yeah, so but mainly I'm speaking about the eukaryotic DNA. Here I'm not speaking about the prokaryotic. They might write it like above the question for you that they are just speaking about the eukaryotic. But if they said eukaryotic, yes, DNA exists in the cytoplasm in a zone called the nucleoid. And DNA can still exist in the cytoplasm in small vesicles called the plasmids. These plasmids are, so let's write it as a note, plasmids are circular pieces of DNA that exist independently of chromosomal DNA. Okay, and these plasmids, they do exist in the cytoplasm, but they just exist for the prokaryotes. Here I'm speaking about the eukaryotes. Okay, now in deoxyribonucleic acid, there are deoxyribose adenine and phosphate radical, deoxyribose guanine and phosphate radical, deoxyribose cytosine and phosphate radical, deoxyribose uracil and phosphate radical, deoxyribose thymine and phosphate radical. So, deoxyribonucleic acid. So, adenine and phosphoric radical, this is correct. Guanine, correct. Cytosine, correct. Uracil, wrong. Thymine, correct. Deoxy means DNA. The only nitrogenous bases that exist are adenine, guanine, cytosine, and thymine. Uracil just exists in the RNA. Okay. Now, in the biochemical analysis of the DNA, always the sum of A and G is equal to the sum of C and T. The sum of G and C is equal to that of A and T. The quantity of guanine is different from that of cytosine. The quantity of adenine and uracil is equal. The quantity of adenine is equal to that of thymine. So, A and G is equal to C and T. What do you think? This is obviously wrong. Okay. Now, G and C is equal to A and T. This is right because G takes C, A takes T. A and T form 2 hydrogen bonds, G and C form 3 hydrogen bonds. The quantity of guanine is different from that of cytosine. This is wrong. The quantity of adenine and uracil is equal. This is also wrong. The quantity of adenine is equal to that of the thymine. This is correct. Why is this wrong? Because uracil does not exist in DNA, it just exists in RNA. The bar body represents a chromosome that has remained coiled, the coiled X chromosome in the male, and an active chromosome, condensed mitochondrial DNA, is sign of nuclear alteration. What is the body of bar? So chromosomes remain coiled. This is correct. The next chromosome in the male, or coiled X chromosome in the male, this is wrong. Inactive, this is correct, obviously. Condensed mitochondrial DNA, this is wrong. Sign of nuclear alteration, this is wrong. The only correct answer is that it's a chromosome that has remained coiled. And it became inactive. This is the bar body. Calcium ions, Ca2 plus ions. So, these ions are important for the movement of cellular organelles, are used for cell adhesion, are used for muscle contraction, are important for exocytosis, are accumulated in the mitochondria. So these are important for movement of cellular organelles. This is so true. Ca2 plus ions are so important in our body for everything. For the movement of the organelles, for the vesicles, for the exocytosis, for the muscle contraction, for cell adhesion, for the cells to stick to each other's. For muscle contraction, this is correct because these Ca2 plus ions, so they entered in the synapse, and then when they enter the synapse, the vesicle, which is filling the neurotransmitters, it starts to go out of the presynaptic membrane to reach the postsynaptic membrane, throwing the neurotransmitters in the synaptic cleft, all because of the Ca2 plus, and then this Ca2 plus will be restored again in this sarcoplasmic reticulum in the muscle contraction. Are important for exocytosis, this is also super correct. Are accumulated in the mitochondria, this is also correct because they have a significant role in metabolism in the mitochondria. Nucleosides and the nucleotides are basic units for the formation of nucleic acids, a system for storing sugars, a possible source of energy, enzymes for the degradation of toxic substances, a source of nitrogen. So nucleosides and the nucleotides, basic units for the formation of the nucleic acids, this is right. A system for storing sugars, this is wrong. Okay, nucleotides, yes, they have the ribose sugar, but it's not a system to store the sugar. The sugar is just stored in a glycogen form to form the glucose, which is 6 carbon. Ribose is 5 carbon, cannot be stored. A possible source of energy, this is correct. Nucleosides and nucleotides can be a possible source of energy since they have some glucose, not glucose, they have pentose sugar, they have some sugar, some kind of carbohydrates inside. Enzymes for the degradation of toxic substances, this is false. Source of nitrogen, this is also false. They can be a source of phosphate because nucleotides and nucleosides, obviously they have a phosphate group, but nothing to do with nitrogen. They have, although they exist in DNA and RNA nitrogenous bases, cannot be a source of nitrogen. Okay, so make sure that you understand the tricks that are existing in these questions. But nucleotides, the main difference between nucleosides and nucleotides, that nucleotides has a sugar, a phosphatic group, and then nitrogenous bases. This nucleotide, however, nucleosides has sugar plus nitrogenous bases, but they do not have a phosphatic group. So the only difference is they do not have a phosphatic group. So here if they added for you, let's say, a question that says contain phosphate, you should say this is also false. Even though nucleotides contain phosphate, nucleosides does not contain phosphate. Okay, now let's go for the fibrous proteins. Fibrous proteins are an integral part of the cytomembranes, constitute collagen, myosin, and microfilaments, are soluble in water, and constitute of the cytoskeletal apparatus, are found only in striated muscle cells, are soluble in water, and constitute antibodies and enzymes. So the fibrous proteins, they're not an integral part of the cytomembrane. Do they constitute of collagen, myosin, and microfilaments? This is true. Are soluble in water and constitute the cytoskeletal apparatus? This is false. Are found only in striated muscle cells? This is also false. Are soluble in water and constitute antibodies and enzymes? This is also false. The concentration of potassium ion is the highest on the outer cytoplasmic membrane, is the highest in the cell cytoplasm, is equal on both sides of the membrane, is the highest in the intracellular fluid, is highest in lysosomes. So potassium, K+, is the highest on the outer cytoplasmic membrane. This is wrong. Okay. Is the highest in the cell cytoplasm. This is correct. Is equal on both sides of the membrane. This is wrong. Is the highest in intracellular fluid. This is correct. Is the highest in lysosomes. This is wrong. So K plus ions, do they exist in the outer of the cytoplasmic membrane? No. Not, they do exist, but in a very small amount. They are so high in the cell cytoplasm, means inside the cell. They're equal on the both parts? No. Usually ions are not equal in the both parts. The fusion, not the fusion, but cellular transport is happening 24 hours. Highest in the intracellular fluid. This is yes, since we already said that it exists in the cell cytoplasm. And highest in lysosomes. No. K+, it's not super high in the lysosomes. Lysosomes are these small vesicles that has hydrolytic enzymes, 31 hydrolytic enzyme, and which are super acidic enzymes. And yeah, here they're trying to trick you in a way that K+ has a plus ion. This means it's losing electrons, it's becoming more acidic, and you would think that it might exist in lysosomes because the inside of the lysosomes, the pH is so acidic. But you should not be tricked. Yeah. In the life cycle of cell, mitosis and meiosis alternate. The phases M, G1, S, and G2 follow one another. Mitosis consists of prophase, metaphase, anaphase, telophase. The S phase is characterized by RNA transcription and protein synthesis. Chromosomes are morphologically demonstrable only in the M phase. So mitosis and meiosis alternate. This is false. The phases M, G1S and G2 follow one another. This is correct. Mitosis consists of pro, meta, and telo. This is also correct. S phase is characterized by RNA transcription and protein synthesis. This is so wrong because the replication of DNA happens in the S. Chromosomes are morphologically demonstrable in the M phase. This is wrong. Okay, because in the M phase it's still not fully demonstrable. Okay, because we start with the chromatin and then becomes a chromosome and then they start to align. So it's mainly demonstrable in the metaphase after the prophase, but in the M phase is immediately after G2, means we're still around G2 pro, between G2 pro. Here we're still in a chromatin structure. We didn't reach yet the condensed morphological structure. In the cell cycle during the G1 phase, the chromosomes are made up of two chromatids. Is this correct? This is wrong. So, no, it's just chromatin. It's nothing to do with chromatids. As the synthesis of lysosomal proteins takes place, this is wrong. In S phase only DNA replications happen. Sorry, false. G0, the DNA is duplicated. This is wrong. G2, the mitotic spindle proteins are synthesized. This is correct. And G2, it starts preparing for M phase. Here the mitosis will start to happen. This means that the two centromeres will start to be far from each others and creating the microtubule, which is the spindle fiber. Okay. Oh my god, I'm sorry. These two are called centrosomes. Or centrioles. Nothing to do with the centromere. Centromere is where the two chromatids touch together, or where the two chromatids accomplish the intersection. Okay, this is the centromere. Here under the centromere exists the kinetochore. So the M cell division takes place. Where did we say? In the cell cycle during the M cell division takes place, the M phase. This is correct. Because the M phase, I'm sorry, why I'm writing wrong. This is correct. This is true. So M cell division takes place. This is fully correct, because here the mitosis starts, so the cell cycle will immediately start. Okay. Stable and perennial labeled cells define the cell types of nervous tissue. Wrong. Lymphocytic cell line. Wrong. The lifespan of the cells constituting the human body. Correct. Okay, because stable and the perennial are the lifespan of the cells inside our body. Okay, it's nothing to do with the nervous tissue or the lymphocytic cell line. We have the lifespan of cells after viral infection, obviously wrong. Phagocytic cell lines, obviously wrong. Histone synthesis. Histones are the proteins that accompany the DNA at the end of the chromosome. Histone synthesis occurs during the G1 phase of the cell cycle, wrong. During S phase, true. During G2, wrong. During G0, wrong. Wrong. Okay. We have DNA synthesis occurs during the G1 phase of the cell cycle, wrong. S phase, true. Wrong. Okay, so in S phase we have DNA synthesis and histone synthesis. Vibratile cilia are an exclusive characteristic of protozoa. This is wrong. Have an exoneme made up of contractile filaments of actin and myosin. This is wrong. Have a fixed portion of made up of a basal corpuscle, plate and roots. This is true. Usually have metachronous movement. This is so true. They are transformed back into centrioles during mitosis. No. So the vibratile cilia are an exclusive characteristic of protozoa. No. Have an exoneme made up of contractile filaments like actin and myosin. Wrong. Have a fixed portion made up of basal corpuscle, plate and roots. Wrong. Correct. Usually have metachronous movement. This is correct. Let's try to highlight the important part. Okay, so this is all about the vibratile cilia. As we know that the cilia is responsible for movement. Okay, cilia and flagella are distinguished from each other by the presence of dynein, the length, the number of them per cell, the constitution of the microtubules, and the mobility. The presence of dynein, wrong. The length, correct, because flagella is longer than cilia. Number of them, yes. Cilia is in a higher number than flagella in a cell. The constitution of microtubules, wrong. The mobility, wrong. Both of them are able to move. Have nothing to do with the mobility. We go back to the vibratile cilia. The vibratile cilia have a basal corpuscle, we just said yes. Are surrounded by a sheath of mitochondria, wrong. Show an exoneme inside them. This is yes, true. Before did they say yes, have an exoneme? Yeah, because they said that the exoneme is made up of filaments of actin and myosin, and that's why we said wrong. But yes, they do have an exoneme. Okay. Are covered by the plasma membrane. This is right. Are characterized by wave-like movements. This is wrong. In the cytoskeleton, microtubules control the migration of exoendocytic vesicles. True. Intermediate filaments are made up of alpha and beta tubulin. False. Tubulin goes for the microtubules. do nothing with the filaments. Intermediate filaments are made up of contractile proteins, false. Actin and myosin constitute the majority of microfilaments, correct. Microtubules have an external diameter of about 25 nanometers. This is correct. So, the intermediate filaments are made up of non-contractile proteins like the keratin, vimentin, desmin, neurofilaments, and lamins. Okay, let's go to the other part, cytoskeleton. The microtubular organizing center directs the organization of cytokeratins, wrong. Okay. The organization of vimentin, wrong. The organization of microtubules, obviously. The organization of nuclear chromatin, this is wrong. The organization and formation of the mitotic spindle fiber, which is right, because it's a microtubule. The energy for the work of shortening the spindle fibers is given by the glycogen of the cell, wrong. ATPase of the cross arms of the microtubule, this is obviously correct. By the glucose that is used during metaphase, this is obviously wrong. By the peroxisomes, false. By the Golgi apparatus, false. It's only used by the energy produced from the ATPase of the cross arms of the microtubules in the spindle fiber. Microtubule formation is hindered by chloramphenicol. This is obviously wrong. Is assembled by interaction with gamma tubulins. This is correct. Okay, because microtubule is mainly made up of gamma tubulins, is inhibited by colchicine, correct. Is also assembled with myosin in case of extreme necessity, wrong. Is assembled with alpha and gamma tubulin, wrong. We said, why I'm writing true on the wrong things? Okay, and wrong. Is assembled with alpha and gamma tubulin, wrong. We just said gamma tubulin. And colchicine usually inhibits the microtubule formation, makes it hard on the microtubules to form. Actin is a protein, component of the cytoskeleton, right? Component of the terminal web, right? Intrinsic of the plasma membrane, wrong. Component of the tubules of the mitotic spindle, wrong. Component of the glycocalyx, wrong. Microtubules contain DNA, no. Participate in the constitution of the centriole, yes. Centrioles make spindle. This is the microtubule. Are of a protein nature, all correct. Have a diameter of 5 to 10 nanometers, wrong. we just said 25 nanometers from some time, are specializations of the apical surface. Wrong. Okay, perennial cells, neurons, or ganglion cells, or smooth muscle cells, pyramidal cells, and plasma cells. Perennial cells, neurons, they can be neurons, true. Ganglions, yes. Smooth muscle cells, wrong. Pyramidal, yes. Plasma cells, wrong.